Hello, this webinar is about the applications of PIXUL Multi-Sample Sonicator for high-throughput sample preparation in multiomics research. We will cover PIXUL applications in different kinds of multiomics research and a variety of samples that have been processed by PIXUL so far. Before beginning the presentation, I would like to ask what challenges you are facing during high-throughput sample preparation. Some of the common issues are listed here. If you have additional challenges or any questions we would like to hear from you, please reach us through here and connect with us on social media, which is provided at the end of this webinar. Previous webinars on PIXUL on the Active Motif website and today's webinar cover how PIXUL addresses all of these issues. PIXUL is being used in high-throughput sample preparation in epigenetic studies involving CHIP, DNA methylation assays, genomic studies, as well as in transcriptomics and proteomics. So PIXUL-based applications transcend through various layers of epigenetics and multiomics research. Despite being part of complicated workflows in multiomics applications, PIXUL is very simple to use, train, and maintain. It is portable and the user does not need earplugs or a cold room to use the PIXUL. PIXUL is ready to use in minutes after powered on. Maintenance is simple, consumables are economical, standard 96-well round bottom microplates is used to run samples. Plates have the option of being tissue culture treated for cells to be cultured in them and sonicated, so there is no sample transfers, hence no sample loss. PIXUL has this intuitive touchscreen through which you can regulate the sonication program of individual columns of a 96-well round bottom microplate. This means that you can lyse different samples in the same run in the same plate if you only program the columns differently. So you save time by not having to wait for sonicating only similar samples in the same batch. Also, it is not compulsory to load samples in all 96 wells because PIXUL allows you to program only the columns in which the samples are loaded. PIXUL performs sonication using the mechanism of cavitation through its transducer lens system. The sophisticated engineering ensures consistent sonication. It also has a Peltier cooling system to control temperature. Now that you have received a basic understanding of PIXUL, I will proceed to give an overview of PIXUL applications in multiomics research. For detailed protocols and applications specific to your research, please reach out to us. PIXUL was introduced in 2019 and its first publication was in nucleic acids research that year. This publication was a collaboration between the University of Washington and Active Motif. Among the authors, Drs. Carol Baumstick and Tom Matula from the University of Washington founded Matchstick Technologies and developed the idea that led to PIXUL. In this publication, PIXUL was used for genomic DNA fragmentation, chromatin shearing for ChIP-qPCR and ChIP-seq, and RNA extraction from mouse tissue for RT-PCR. These are the different samples that were processed by the PIXUL, and the protocols included the following products from Active Motif. One of the experiments in this publication shows ultrasound treatment efficiency and consistency in genomic DNA fragmentation by PIXUL. Here, 100 microliter of salmon DNA was loaded in each of the 96 wells in the microplate, and the total time of sonication for the entire plate was only 36 minutes. The results represented in the waterfall plot here show efficient and consistent shearing sizes of sonicated DNA. And the sonicated DNA sizes are shown in this x-axis here. This waterfall plot and these bar graphs are derived from the 12 wells of an individual row of the 96 well plate. to underscore the efficiency and consistency of the pixel. The bar graphs represent band fraction per well in the 200 to 600 base pair ranges. One of the other experiments showed that PIXUL can be used for multi-purpose sample preparation for integrative studies like chip qPCR and RT-PCR. Here, PIXUL was used to sonicate chromatin from these 4 mouse tissues to perform chip for RNA polymerase II, and also to extract RNA from those tissues for RT-qPCR. Consistently, the chip results corresponded with the RT-qPCR results, as you can see here. And this establishes the use of PIXUL in multiple workflows for integrative research in multiomics. Another publication in 2019, focusing on diabetes and cardiovascular disease risk, used PIXUL in a chip experiment on mouse liver. This was a collaboration between different universities and the pharmaceutical industry. A very recent publication on PIXUL has integrated it in an assay for the epigenetic modification of DNA methylation called MEDIP, or methylated DNA immunoprecipitation. One of the applications of PIXUL in this study is the sonication of induced pluripotent stem cells, or iPSCs. And that was done for ChIP-seq to analyze the histone H3K27 or lysine 27 trimethylation. This modification is an epigenetic mark associated with transcription repression. The study focuses on identifying critical functional sites of PRC2, which is the polycomb repressive complex 2 that catalyzes H3K27 trimethylation. So in this direction, a computationally designed protein called EED binder, or EB, which inhibits the PRC2 function, is fused with DCAS9 to generate EBDCAS9. And this causes PRC2 inhibition at a precise locus using guide RNA. So here in these 2 panels, we see the ChIP-seq analysis of histone H3K27 trimethylation for genome-wide and for developmentally regulated genes. This study also presents integrative analysis of ChIP-seq and ATAC-seq results, which are not shown here, but those results once again demonstrate how PIXUL helps high-throughput workflows for integrative multiomics analysis. Since we are talking about ChIP-seq, so in that context, I would like to briefly draw your attention to another webinar on our website, which is shown through this link here, where PIXUL sonication of 10 human tissues and 7 mouse tissues are shown. And this includes complicated samples like PBMCs, as you can see here. So now coming back again to the Cell Reports publication, as mentioned earlier, the other new application for PIXUL here is DNA methylation called MEDIP. So in this assay, single-stranded DNA is isolated from iPSCs and then sonicated in PIXUL for 6 minutes, which was subsequently subjected to immunoprecipitation using 5-hydroxymethylcytosine or 5-HNC antibodies. And the regions for PCR are shown here, which was upstream of the transcription start site, or TSS. The MEDIP assay showed that EBDCAS9 induces an increase in hydroxymethylation, which is actively involved in DNA demethylation and transcription activation. So in summary, the study presents that EBDCAS9 causes epigenomic spreading and recruits transcription activation marks, and a PRC2 controlled TATA box was identified upstream of the transcription start site of TBX18, which is a gene that is critical for development. Now, very recently, Active Motif has developed a novel method to study DNA methylation called ABBS, or anchor-based bisulfite sequencing, published in Nature Communications Biology. And a webinar on ABBS is also available on our website. So in this publication, human and bacterial samples were processed in PIXUL, and the following products were part of the protocol. In ABBS, genomic DNA from 3 microgram of E. coli or 10 microgram of human K562 cells was spiked with 1% of unmethylated lambda DNA followed by bisulfite conversion. The detailed methods are given in the paper. The DNA was then eluted and sonicated using PIXUL for 15 minutes, followed by the rest of the protocol as outlined here. The experiments on bacterial and human cells show that ABBS provides increased coverage of 5-MC or methylation of the 5th carbon of cytosines. So overall, PIXUL helped with enhancing the speed, consistency, and throughput of sample preparation in this publication. And it also shows that you can process a range of samples starting from bacteria all the way up to humans. Now moving on to other areas of multiomics, this preprint in bioRxives presents the integration of PIXUL in transcriptomics or RNA sequencing workflow using human cell culture and mouse tissues. The workflow is represented here. After the tissue samples were obtained and loaded onto the 96-well plate as shown, PIXUL was used for 30 seconds only to process the full plate and to obtain the RNA, thereby greatly reducing the time to work with high-throughput samples. The RNA-seq profiles are shown here, and the red dashed lines and the purple dashed lines are the samples prepared with different buffers, and the green and the blue peaks here are the experimental and controls respectively. Overall, PIXUL helped with reducing the time, labor, and enhancing the consistency and throughput of the transcriptomic workflow. We now move on to another aspect of multiomics, that is proteomics. PIXUL has been used to process a variety of samples for proteomics applications involving mass spectrometry. Here we show the buffer and PIXUL processing time for human cells, yeast cells, and tissue. It's interesting to see that despite their tough cell walls, yeast was processed in PIXUL without needing additional lysis by any other methods. For detailed protocols and information on additional sample types processed by PIXUL for proteomics, please reach out to us. The final application slide for today is recombinant protein extraction from bacterial cultures using PIXUL. The bacterial cultures containing 9 gene expression constructs were grown and induced, and then the cells were sonicated in PIXUL for 3 minutes only to extract the proteins. And you can see the gel image here for that experiment. The detailed protocol is available in the documents section on our PIXUL webpage. So in summary, I hope that you are convinced about the features and advantages of PIXUL, that PIXUL can process various types of samples. It can be very quick and high throughput and consistent just based on the way the transducers are built. And it is very versatile because you can program different columns. You can program from column 1 to column 12 individually. And then it is very user-friendly, easy to train and maintain. And in the long term, the consumables and the equipment is quite economical. So I would like to encourage you to please reach out to us and connect with us on social media for our offerings and promos. And please stay tuned for more webinars on PIXUL and our other portfolio. We look forward to hearing from you. Thank you very much for listening to this webinar.